Hello, everyone, and thank you for attending today's webinar, Amplicon Sequencing and Introduction. To give an overview of what we'll be talking about today, we will first discuss what Amplicon sequencing is, then how Amplicon library preparation is different from whole genome library preparation. We will cover which kits and workflows can be used to prepare Amplicon libraries. We will look at what an Amplicon sequencing run looks like. We'll discuss how low base diversity can affect a sequencing run. What's the best ways to sequence low diversity Amplicon libraries? And what analyses are available for Amplicon data? So first, what is Amplicon sequencing? Amplicon sequencing is a method of targeted sequencing to look at a specific region, locus, or set of regions. It lets us get a closer view of specific genes or targets of interest. It allows for deeper sequencing of these regions, resulting in higher sensitivity. And it is more cost-effective and time-efficient than other types of sequencing, such as whole genome sequencing, when we are interested in only a specific region. Applications of Amplicon sequencing include cancer, forensics, inherited disease, agrogenomics, metagenomics, and more. Now let's compare whole genome sequencing libraries with Amplicon libraries. First, we will look at a genomic DNA library preparation. We start with genomic DNA, and we randomly fragment it into smaller pieces, creating many unique fragments. And then we add adapter sequences to create a library ready for sequencing. This results in a library with many different unique inserts, which yields a balanced or diverse library because at any given position, we expect roughly equal proportions of A's, T's, C's, and G's. You can see at this single position, each base is present and balanced. Let's contrast this with an Amplicon library prep. In this method, we start with genomic DNA and selectively amplify a single region and then add adapters. This results in a library where the inserts are largely the same sequence, creating a very low base diversity library. You can see at a given position, only a single base may be represented. These examples represent the far ends of the spectrum from the highest to the lowest in diversity. You can imagine if we had a library composed of multiple amplicons, it would increase the overall diversity of the library, but would still not be as diverse as a genomic library. And we'll see an example of this a little bit later. Now, which kits or workflows can we use to prepare Amplicon libraries? We will discuss 3 methods today for Amplicon library generation, and after each, I will include some resources for more information. The first workflow is AmpliSeq for Illumina. AmpliSeq for Illumina allows for both custom and fixed panel options, and it is compatible with low input integrated samples. The second workflow we will discuss is the Nextera XT and Illumina DNA Prep and Tagmentation library preparations. For these, PCR amplicons are prepared in advance, and random fragmentation is used during the preparation, which adds base diversity to the libraries. The third workflow is the Illumina demonstrated 16S and internal transcribed spacer, or ITS, protocols. This is a 2-step PCR workflow. It has a single target region per library and can be customized for novel targets. Our first workflow is AmpliSeq for Illumina. We will start with our input material, which may be DNA or RNA or both, depending on the panel. Then, primer pairs will create multiple amplicons across the region or regions of interest. The primer sequences are removed, then we ligate adapters and amplify the library fragments with PCR. After a cleanup, the libraries are ready for QC and sequencing. When you are validating your AmpliSeq for Illumina libraries on the fragment analyzer, file analyzer, or other trace instrument, the distribution that you will see will depend on the panel content and amplicon size. These are 2 examples of AmpliSeq libraries. For ready-to-use panels, there will be an example trace in the reference guide for the expected library distribution. We currently have the option to pull up to 384 samples per run using index sets A through D. Panels contain multiple Amplicon targets, which add diversity to the library. There are DNA and RNA panel options. Input can range from 1 to 100 nanograms, and we typically recommend 10 nanograms of input material per pool for high-quality input. With lower quality samples, we may want to increase the input material if possible. We have 12 ready-to-use panels for use with DNA input, RNA input, or both, depending on the panel, and we're continuing to build our AmpliSeq portfolio. We have additional community panels available, which have been created by key opinion leaders in their field. Some examples of the available community panels include Canadian Inherited Cancer Panel, Antimicrobial Resistance Research Panel, and Autism Research Panel. We can also create custom DNA AmpliSeq panels using Design Studio, targeting not only the human genome, but other species as well. Design Studio can also be used to create custom RNA panels for the human genome. There are options for RNA expression, fusion, and targeted RNA panels. I'll give a brief overview of the custom AmpliSeq design process. First, in Design Studio, we will start a new design. If we choose RNA as the assay type, we'll have three options. Expression, which is a single pool design for targeted gene expression analysis. Fusion, which is for targeted gene fusion panels. And TruSeq targeted RNA, which is a sequencing-based gene expression assay, which allows for up to 1,000 targets in as many as 384 samples on the MiSeq. Please note that these options are only available for the human genome. If instead we choose DNA as the assay type, then we have four AmpliSeq options. Gene, which is useful for covering full genes or regions of interest. Hotspot, which allows for designing panels that target SNPs and small indels. On Demand, which allows you to select from predetermined wet lab-tested primer pairs. And Enrichment, which allows for targeting large gene panels for use of Illumina DNA Prep with Enrichment Library Preparation Kit. We can then choose the species we want the panel to target from these options. We can then configure more specifics about the design, choosing the sample type and the Amplicon length. For regular input, we can choose one of four Amplicon lengths, whereas with FFPE and cfDNA or cell-free DNA options, the Amplicon length will be limited to smaller sizes to ensure good panel performance. Once we've configured the design parameters, we can add the targets we would like to cover by gene name, chromosomal coordinate, or we can use a template to upload multiple targets at once. We can also copy Amplicons from pre-existing designs or from community and fixed panels. The minimum number of Amplicons for a design is 12, while the maximum is over 12,000. After all of our targets have been added, we submit the design. Once the design completes, we can review the coverage and further optimize the design if we would like. Click See More Solutions under the pie chart to be able to see designs made with different stringencies, which may have improved coverage. We have detailed recorded webinars that discuss AmpliSeq for Illumina and Design Studio. I've also included links for our ready-to-use AmpliSeq and Illumina panels, our community panels, and Design Studio for creating your own custom panel. Now let's discuss how to use the Nextera XT DNA Library Preparation Kit to prepare Amplicon libraries. First, you will need to design and create Amplicons you are interested in sequencing. Once we have the PCR Amplicons, we mix them with the Nextera transposomes. The transposomes tagment them to fragment and add the tagging sequences to the ends of each fragment. We then use PCR to add the remaining adapter sequences and uniquely index each sample to complete the library. When checking Nextera XT libraries, we typically see a broad distribution of fragment sizes. The bioanalyzer traces here are from genomic DNA input. With Amplicon input, the input DNA will be smaller, so we can see smaller final library sizes and may see more narrow distributions. For the Nextera XT library preparation, you will need 1 nanogram of input material, and this can be 1 amplicon or multiple amplicons. Amplicons will need to be 300 base pairs or larger. To ensure good coverage of the complete target region, we recommend adding an extra 50 base pairs to either side. Library preparation will require the Nextera XT library preparation kit and 1 or more indexing kits. With multiple indexing kits, we're able to pull up to 384 uniquely indexed libraries per sequencing run. And the random enzymatic fragmentation in the workflow adds base diversity to the final libraries. For more information about this workflow, you can refer to the support page. We also have a great recorded webinar available at the link here. The Illumina DNA Library Preparation Kit, formerly known as Nextera DNA Flex, is also an option for Amplicon library preparation. While the Nextera XT and Illumina DNA prep workflows are similar, there are some key differences to consider. To prepare libraries with the Illumina DNA prep kit, we combine the DNA amplicons with bead-length transposomes to tag and fragment the amplicons. We then finish the library preparation by uniquely indexing and completing the adapter sequences using PCR. This is a bioanalyzer trace for an Illumina DNA prep library prepared with genomic DNA. The Illumina DNA prep workflow does include a bead-based size selection step after the indexing PCR, so depending on the size of your input amplicons, you may need to adjust the final size selection, and this would lead to a shift in the distribution towards a smaller average size in base pairs. The Illumina DNA prep kit allows for flexibility for the amount of input material. Similar to Nextera XT, single or multiple amplicons can be used. Illumina DNA prep can be used with amplicons 150 base pairs and larger. As with Nextera XT, we recommend adding an additional 50 base pairs to either side of the region of interest. Library preparation requires the Illumina DNA prep library preparation kit and the Nextera DNA combinatorial dual, or CD, index kit, or the IDT for Illumina DNA/RNA UD index kit. And we can pull up to 384 uniquely indexed libraries with the IDT for Illumina DNA/RNA UD indexes for sequencing. We also have a recorded webinar with useful information for the Illumina DNA prep workflow. The support page linked here includes protocol information and frequently asked questions. The final workflow we will be discussing today is the Illumina demonstrated Amplicon workflow, 2 applications of which are the 16S and ITS metagenomics protocols. This is a 2-step PCR workflow. In the first PCR, we use locus-specific primers with overhangs to amplify a single region of interest. Then we use a second PCR, which utilizes the overhangs to add the rest of the necessary adapter sequences and uniquely indexes the samples to complete the libraries. And then we're ready to QC the libraries and proceed to sequencing. When checking your libraries, you will see a discrete peak. As the libraries target a single amplicon, the distribution will typically be very narrow. So you can see variability if you are targeting a variable length locus or the same locus in a mixed species sample. If we follow the ITS demonstrated protocol, this protocol uses the pool of amplicon primers in the first PCR, targeting the ITS1 region and fungal species. This results in 2 main peaks, which can vary slightly in size relative to the taxa. For the demonstrated Amplicon workflow, each library targets a single region for amplification. It requires 12.5 nanograms of input, and uses the Nextera XT indexing kit to allow for pooling up to 384 samples per run. You can design your own locus-specific primers for the first PCR to target novel regions. This workflow is very similar to many published protocols for targeted Amplicon sequencing. However, using this workflow ensures that your libraries will be compatible with any Illumina sequencer without any custom settings or custom sequencing primers. There are a few considerations for designing a locus-specific primer. First, we should target an Amplicon that will have about 50 base pairs of overlapping sequence when sequenced. For example, if running a 2 by 300 base pair paired end read, Illumina recommends having an insert size of 550 base pairs or smaller so that the bases sequence at the end of each read overlaps. Second, the locus-specific portion of the primer must have a melting temperature of 60 to 65 degrees Celsius. For the melting temperature calculation, only use the locus-specific portion of the primer, but for hairpin and dimer calculations, use the full sequence, including the overhang. Lastly, we recommend using standard desalting purification when ordering oligo primer sets. Here is a link to the Illumina 60NS demonstrative protocol and our fungal metagenomics ITF demonstrative protocol. This is a quick overview table of the 4 preparation methods that we've discussed as a reference, and I'll leave this up for a couple of moments for you to skim over the table. Now that we've discussed how to prepare libraries, let's proceed to sequencing, and we'll take a look at what an Amplicon sequencing run looks like. First, let's discuss the 3 main metrics we'll be considering to evaluate these sequencing runs. The first is percent base. This will tell us the percentage of A's, T's, C's, and G's in each cycle of the sequencing run. The second metric is the percent passing filter, which tells us the percentage of the clusters on the flow cell that are giving a pure signal and can be used to generate sequencing data. The third metric is the Q-score, and we will be looking at the percentage of base calls above Q30, which is synonymous with a 99.9% confidence in the base call. The higher the percentage at or above Q30, the higher the overall run quality. First, let's look at the run metrics for a balanced, diverse library. These are the metrics from a PHI-X run, which is a viral genomic library. This is the percent base plot. On the x-axis, we have the cycle and the sequencing run, and on the y-axis, we have a percent scale. Each of the 4 bases is represented by a color, and each has its own line. When we look at the percent base, we see fairly equal representation of each base in each cycle, and the base percentages are even throughout the run, indicating that the bases are balanced. We can then look at the percentage passing filter. The blue whisker plot represents the raw cluster density. In this graph, we see it's about 700 K per millimeter squared. The green plot indicates the cluster's passing filter. The green plot is very close to the blue plot, which indicates a high percentage of the raw clusters are also passing filter. For this particular run, the passing filter is just over 95%. Now let's look at the Q30 for the run to evaluate the quality. On the x-axis, we have the Q-score. The higher the Q-score, the higher the quality. On the y-axis, we have the number of reads in millions. A Q-score of 30 or above is considered high-quality data. You can see that it is high for this run at around 90%. This next run is for an AmpliSeq for Illumina panel sequenced on the MySeq using V3 chemistry. Each library includes approximately 4,600 different Amplicons. When we look at the percent base plot, we see that it is spiky, with one base having a higher percentage than the other bases at a given cycle, and this skew towards one base changes from cycle to cycle. Because the panel is made up of a mixture of amplicons, the overall diversity is much lower than the previous example of the randomly fragmented genomic DNA library of PHI-X. We can now look at the clusters passing filter. The green box clusters passing filter is quite close to the blue, which is the raw density. The clusters passing filter for this run is about 82%. And last, we look at the quality for the run. We see that our Q30 percentage is at 87.6%, which is quite high and tells us that the data quality is good. This last run is a 16S library preparation on an ATCC, or American Type Culture Collection, microbiome standard on the MiSeq using V3 chemistry. Each library is a single amplicon of the variable V3 and V4 regions of the 16S rRNA gene. Different bacterial species will have slightly different sequences for this region. When we look at the percent base plot, it is extremely spiky, with one base reaching as high as 90% of the signal at a given cycle. Such low diversity is typical of sequencing a single amplicon, which has very little variation between the samples. Runs with such very low diversity samples can benefit from a PHI-X spike-in to introduce base diversity. In fact, this run includes about a 10% PHI-X spike-in, which is why the maximum signal from a given base is around 90% rather than 100%. We can now look at the clusters passing filter. This run had a raw cluster density of about 600 K per millimeter squared, lower than the typical density of 1200 to 1400 K per millimeter squared for V2 runs, to help mitigate the effects of the low diversity. Since there are fewer clusters that are well spaced apart, the clusters passing filter is around 90% for this run. We will review this concept more later in this webinar. And last, when we look at the quality for the run, we see that our Q30 percentage is at 70.1%, which is still quite high and tells us that the data quality is good. Now that we have seen what amplicon sequencing runs look like, let's discuss how low diversity can impact a sequencing run. The first impact low diversity can have is on the template generation step. On non-patterned flow cells during template generation, the sequencer creates a map of the position of each cluster on the flow cell. If there are multiple bases present, it is easy to distinguish each cluster and make an accurate map. With low diversity sequencing, at a given cycle, only a single base may be present. So clusters that are close together showing the same base may be seen by the sequencer as a single cluster, creating an inaccurate cluster map. The second impact is on matrix correction. Matrix correction is how the sequencer accounts for the crosstalk between the fluorophores and correct intensities. You can see from the emission plot that there is spectral overlap in the intensities that come from the bases, so we have to correct for this to confidently distinguish each base. If the majority of clusters are all showing the same base in a given cycle, the intensity baselines will be skewed, which will impact the matrix correction. If we have a poor map from template generation and inaccurate matrix correction, then the sequencing software will not be able to make a confident base call, which will result in low quality data for the run. For more information about template generation, matrix correction, and other considerations for low diversity sequencing, please see the recorded webinar available at the link here. Now that we've discussed how low diversity can impact our run, let's cover the best way to sequence low diversity amplicon samples. We want to mitigate the low diversity of libraries in order to improve data output and quality. If we have higher diversity amplicon libraries, we may not need to use the upcoming strategies to improve the run. If you have any questions about the best way to sequence your amplicon libraries, please feel free to reach out to technical support. The first way we can improve low diversity amplicon sequencing is to spike in the well-balanced genomic library PHI-X. For a well-balanced run, we recommend spiking in 1% of PHI-X as a sequencing control. For low diversity sequencing, we can also use PHI-X to add base diversity at each cycle. On the MiSeq using V3 chemistry, we recommend spiking in between 5 and 10% PHI-X into your library pool. We want at least 5% aligning during the run, so you may need to spike in a higher percentage to ensure that 5% aligns. Spiking in PHI-X will increase and improve the base diversity in the run. It will improve the Q-scores for the quality for the run, and it will increase the percentage of clusters passing filter, which corresponds to an increase in data output. Please note, if we are sequencing tagmented amplicons, we don't need as much PHI-X due to the random fragmentation of the transposons. The PHI-X spike-in percentage will depend on the instrument you are using. We have a helpful bulletin which outlines the recommendation for each sequencer here. The second way we can improve low diversity sequencing is by lowering cluster density. For example, for a well-balanced library sequenced on the MiSeq with V3 chemistry, we typically want to see a cluster density of 1200 to 1400 K per millimeter squared. For a low diversity run, we want to lower that to between 700 and 1000 K per millimeter squared. Cluster density may be adjusted in subsequent runs to maximize quality and data output. If we reduce cluster density, each cluster has more space, allowing for a more accurate map and sequencing. Lowering cluster density will not change the percent base, but it will improve the quality of the run and increase the clusters passing filter, so it will increase the output of the run. Once we have our sequencing data, how can Amplicon data be analyzed? There are both BaseSpace and local analysis options available for Amplicon datasets. On BaseSpace, if our input is DNA, we can use the DNA Amplicon app. If our input is RNA, we can use the RNA Amplicon app. If our input is both DNA and RNA, instead of launching each app separately, we have a combined DNA plus RNA Amplicon app that will run both analyses. For local analysis, we can use Local Run Manager, either the on or off instrument versions. LRM is designed for benchtop sequencers, including the MiniSeq, MiSeq, NextSeq 500, 550, and iSeq 100. It is not designed for the NextSeq 1000, 2000, HiSeq series, or NovaSeq 6000. Similar to the BaseSpace apps, if we have DNA input, we can use the DNA Amplicon module. If we have RNA input, we can use the RNA Amplicon module. And if we have DNA plus RNA samples, we will need to use those modules independently. For more information and support for BaseSpace apps or LRM and its modules, please see these links. I'll now give a quick summary of our talk today. We discussed that Amplicon sequencing is a method that gives us a closer look at specific regions of interest in the genome. Amplicon libraries are lower in diversity than other library types, and some are very, very low in diversity. We discussed 3 methods to prepare Amplicon libraries: AmpliSeq for Illumina, Nextera XT and Illumina DNA Prep, and the Illumina Demonstrator Protocol, 2 applications of which are the 16S and IPS metagenomics protocols. We reviewed Amplicon sequencing metrics, focusing on the percent base, a measurement of nucleotide diversity, clusters passing filter, which is a measure of the percentage of the clusters on the flow cell that give a pure signal and can be used to generate data, Q-scores, the overall measurement of data quality. We discussed 2 strategies that we want to use to improve low diversity Amplicon runs. The first is spiking in PHI-X, and the optimal spike in percentage will differ depending on the sequencer and chemistry you are using. The second is reducing the cluster density. We can analyze Amplicon data with BaseSpace or Local Run Manager, use the DNA, RNA, or DNA plus RNA Amplicon apps on BaseSpace, and use the DNA or RNA Amplicon modules on LRM. I have some additional resources for you as well. We have public demo data sets, including the 16S and AmpliSeq panels on BaseSpace. which are a good reference for sequencing run metrics and sequencing analysis. We also have the Library Prep Kit Selector, which is helpful if you're unsure of the best method to use for your project. The Bulletin Board, which has helpful documents on a variety of topics. NGS for Beginners page, which has a variety of resources for getting started with NGS. And the main support page. We also have online recorded webinars and upcoming webinars, and you can always contact technical support with any questions. Thank you so much for taking the time to join me today. I hope that this session has been helpful to you.